So we start with the topic structure and types of chromosome. So in this particular figure, the structure of the chromosome is seen. A chromosome has two arms separated by centromere. Okay, so it has got a short arm as you see in the figure, and long arm, and there is a centromere. Okay, the ends of the arm are called as telomeres. The arms are divided in two, and each part is known as chromatid. Okay, now again, there will be pictorial questions. They will show you the uh, picture of a chromosome, and they'll ask you, there will be some X or Y, and what X denotes, what Y denotes. So it may be centromere, it may be telomere, etc., etc. So you should know the parts of the chromosome. Okay. Second, we'll go through the important terminologies. So what is chromatid? Each of the two identical halves of a replicated chromosome is called as chromatid. So sister chromatids are joined at the centromere. Okay. What is centromere? The constricted region of a chromosome where the sister chromatids are held together and where the spindle fibers attach during the cell division. Okay, these are also SBAs. Okay, so they'll give you some description. They will ask you what it is, whether it is chromatid, whether it is centromere. Okay, so coming to the telomere, the repetitive nucleotide sequences at the ends of the chromosome that protect them from deterioration or fusion with other chromosomes. So that is what is the function of the telomere. Okay, so that is also SBA. What is the function of the telomere? So it protects them from deterioration or fusion with other chromosomes. Okay, again there is a SBA on kinetochore, a protein structure on the centromere that attaches the chromosome to the mitotic spindle during the cell division. Okay, so they'll give you the description that the structure important, a protein structure on the centromere that attaches the chromosome to the mitotic spindle during cell division is called as, and they will give you the option chromatid, centromere, telomere, kinetochore. So it is the kinetochore, which is a protein structure on a centromere that attaches the chromosome to the mitotic spindle during the cell division. Okay, now coming to the histones. This is protein around which DNA binds to form nucleosomes, the basic unit of chromatin. The nucleosome, a structural unit of chromatin consisting of a segment of DNA wound around a core of histone proteins. Then you have euchromatin, lightly packed form of chromatin that is transcriptionally active. It contains genes that are actively expressed. So euchromatin is a lightly packed form of chromatin. Remember, and heterochromatin is a densely packed form of chromatin. So there is a difference between euchromatin and heterochromatin. So euchromatin is a lightly packed form of chromatin that is transcriptionally active, while heterochromatin is a densely packed form of chromatin that is transcriptionally inactive. Okay, then you have P arm and Q arm. So P arm, that is the shorter arm of chromosome, named after the French petit, meaning small. Okay, and Q arm, that is a long arm. The long arm of chromosome is named after Q as it follows the P alphabetically. Okay. Then homologous chromosomes. Pair of chromosomes, one from each parent, that is similar in shape, size, and gene content. They are called as homologous chromosomes. Then you can, what is aneuploidy? A condition in which there is an abnormal number of chromosome in a cell. So trisomy 21 in Down syndrome. So there will be 21 chromosomes. Okay. Then you have polyploidy, a condition where the cells have more than two complete sets of chromosomes. Normally, there are only two sets of chromosomes, each with a 23. Okay, that forms 46 chromosomes. But sometimes there may be three sets or four sets of 23. Okay, that is called as polyploidy. Then structural operations, changes in chromosome structure such as deletions, duplications, inversions, translocations, etc. Then chromosome territories, specific regions of the nucleus occupied by individual chromosomes during the interphase. Okay. So what are chromosomes? In eukaryotic cells, within the nucleus of each cell, the DNA is found in the compartments in the form of numerous linear molecules, which are together with certain proteins make up the chromatin. When the cell starts to divide, this chromatin condenses to form chromosomes. Each chromosome is made up of DNA, which is tightly coiled many times around a protein called as histones that support the structure. Chromosomes were first described by Strasburger in 1815, and the term chromosome was first used by Walder in 1888. They appear as a rod-like dark stained bodies during the metaphase stage of mitosis, and when cells are stained with a suitable basic dye and viewed under the light microscope. Again, here SBA can be asked in which stage they can be viewed. So they can be seen in metaphase stage of mitosis under the light microscope. Okay, so in this particular phase, metaphase stage of mitosis, they can be seen under the light microscope. So coming to the types of chromosome, again they can ask you a pictorial diagram, and they will ask you this type of, which is the type, this type of chromosome. So depending upon the centromere, it is classified as a telocentric rod-like chromosome with centromere present on the proximal end. No p-arm or the short arm of the chromosome is present. So first figure you see is a telocentric, where there is no short arm. Okay, so telomeric chromosomes are not found in the humans. Okay, so telocentric chromosomes are not found. And where is the centromere? The centromere is present on the proximal end. Then second is acrocentric, rod-like centromere present at one end, giving rise to one very short arm and exceptionally long arm. So second, you see a acrocentric. Okay, so this is upon the depending upon the position of the centromere. Third is a submetacentric. It is L-shaped or J-shaped with centromere near the center of the chromosome. It is not at the center, but it is near the center of the chromosome, giving rise to two unequal arms. Okay. And third one is a metacentric. It is a V-shaped chromosome with centromere in the middle, giving rise to two equal arms. Okay. So that is what is the classification depending upon the location of the centromere. Then on the basis of number of centromeres, so monocentric, which has got only one centromere; dicentric, which has got two centromeres; polycentric, more than two centromere, it is a polycentric. Then acentric, without any centromere. Such chromosomes represent freshly broken segments of chromosomes which do not survive for long. Then fifth is a diffused or non-located with indistinct centromere, diffused throughout the length of the chromosome. Okay, so these are the types of chromosomes. Then another classification is the autosomes and sex chromosomes. This we have already seen. Human chromosomes are of two types. One is autosomes and second is a sex chromosome. Genetic traits that are linked to the sex of the person are passed on through the sex chromosomes. The rest of the genetic information is present in the autosomes. Okay, humans have 23 pairs of chromosome in their cells, out of which 22 pairs are autosomes and one pair of sex chromosomes, making a total of 46 chromosomes in each cell. So this is what is the 23 sets of chromosome. 22 are autosomes, and the last pair which you see is a sex chromosome, and it may be XY or it may be XX. Okay. What are the functions and significance of chromosome? It is a genetic code storage. Second is a sex determination. We know the X chromosome, when two X chromosomes will be present, it will be female sex will be get formed, and if there is a Y chromosome XY, then the male sex will be formed. Then control of cell division and formation of proteins and storage. These are the functions of chromosomes. Okay, so that finishes the topic of structure of the chromosome. Thank you.